Welcome AP Bio students to your review of Chapter 14 and 15, Basic Genetics. I'm going to review a few things that I know will be on your test tomorrow, and this might be a good idea to look over this before your AP exam, which is coming up shortly here. What we're going to be looking at in this video are Mendel's laws and basic Punnett squares, and then going beyond Mendel's laws. I've also got some other websites I'd like you to check out that'll be helpful in studying for this test that you have coming up. Mendel's laws. So I felt like this was a little confusing to some, and I wanted to review real quickly the law of segregation and the law of independent assortment. Segregation, basically I feel like the only reason it's confusing is because people try to overthink it. Really what segregation is trying to describe is what happens during meiosis, which of course Mendel didn't know anything about, but segregation basically means that every adult carries 2 alleles for every gene. So for all 30,000 of your genes, an adult can carry up to 2 of each gene. But when they go to make sex cells, they can only give that sex cell 1 of every gene. So 1 allele makes it into each sex cell. So that's really what my second bullet point is stating here. So individuals are only giving 1 allele to every sex cell, meaning that each offspring gets 1 allele for each gene from each parent. Getting back to the diploid number so that every human child ends up with 46 chromosomes. Mendel's second law, the law of independent assortment. That's the one that states that the inheritance of 1 allele has no bearing on the inheritance of an allele of a different gene. And law of independent assortment almost always holds true. Realistically, chromosomes assort independently, and most genes do as well. Even if genes are on the same chromosome, they should assort independently because of how common crossing over events are during metaphase 1 of meiosis. So that's something to consider. That independent assortment really does hold true. The discrepancies with independent assortment, where independent assortment starts picking up, I don't know, trivial little problems with it, that's what led Morgan to later stumble on gene mapping, because he mapped out the occurrence of crossing over events using Mendel's law of independent assortment and where it sort of falls short. Make sure you understand that relationship. Ooh, what else we got? Basic Punnett squares. Please make sure you can do a basic Punnett square. Flaming rabbits. So what's going on here is we have 2 purebred bunnies. This is just going over a basic Punnett square, making sure we all follow what goes on in a basic Punnett square. Both of these bunnies are purebred, as you can see. The black one, that's going to go on the left side of the Punnett square, he is purebred for the dominant version of hair color, which would be black, and the white one is purebred for the white version, the white allele of the hair color gene. And when we cross these 2 P generation bunnies, we end up with all heterozygous offspring in the F1 generation. And those would all be black because dominant is completely dominant over the recessive allele, and you end up with these cute little delicious looking hybrids. Hooray. If we were to then breed those hybrids together, we would end up with Mendel's 3-to-1 ratio that he was predicting. As far as phenotypic ratios go, you'd end up with a 3-to-1 ratio, 3 black bunnies and 1 white bunny. And if you're looking at genotypic outcomes, you would end up with 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive offspring if you did an F1 cross. I would know terminology like P generation, F1, F2. All of that is fair game for test questions. Oh, what else do I need to tell you here? We looked at several inheritance patterns that are more complex than just basic Mendelian inheritance. Basic Mendelian principles like basic Punnett squares do describe how most genetics work. 90% of the genes in humans are going to be just basic Mendelian genes, but there are some non-Mendelian genes that you're also going to need to know about are non-Mendelian inheritance patterns. We talked about polygenic traits. Polygenic traits, realistically, each gene that makes up the polygenic trait is a Mendelian gene. But then you just multiply the outcomes of the little Punnett squares get multiplied together to give you the overall probability of a polygenic trait. And it results in a bell curve of phenotypes. So that's where we get the bell curve distribution of skin color, hair color, eye color in humans, because those are all polygenic traits. Incomplete dominance, I keep using the Snapdragon example in class where you have the red flower and the pink flower. No, the red flower and the white flower, and when you cross them you end up with pink offspring, which is non-Mendelian, because if it was a Mendelian gene, the dominant trait would completely cover the recessive trait. But with incomplete dominant genes, you end up with some of the recessive gene showing through. Epistasis deals with epigenetic pathways, basically that changing one gene changes the expression of other genes in the DNA. You'll need to know epistasis. Pleiotropy is one gene that controls multiple phenotypes. We talked a little bit about gene linkage and sex-linked traits and how their inheritance pattern is different. Sex-linked traits are going to, the phenotype will show up in males and be carried by females, because females have a second X chromosome that can act as a backup, whereas males don't. So if they end up with a genetic abnormality in an X chromosome and they only have one X chromosome, they're going to have to display the genetic abnormality in their phenotype. Finally, I have some other resources that I think would be a good idea to check out. This guy, Bozeman Science, this is his high school. He's an AP Bio teacher who he's doing some of the same things I'm doing, but he's much, much better at it. The top link is just to his general site. If you ever have any biology questions, he's got a video on just about everything on Earth, and he's a very good teacher. The bottom link there that has the long extension, that's specifically for a genetics video that will help you specifically on this test. But BozemanScience.com is it's an excellent resource. I highly suggest you look at his stuff. Finally, there's Gregor Mendel. That's really everything I've got for you. Of course, all of your reading is fair game. I would make sure that I've read the chapters in the textbook. If you have any questions, feel free to email, and I'll see you soon. Good luck studying.